This is the restriction enzyme digest lab. And here we're going in the prediction section because we already read the context and material section. It says during this simulation, we'll be performing a restriction digest using BAM-HI and HIN-3 onto plasmids, the power and the pKnR. So here the current structure and size. As you can see, the pKnR has 5,512 base pairs, and the para has 4,872 base pairs. So based on this plasmid math, how many fragments do you expect to create if using both BAM-HI and HIN-3? So I expect each of them to have 2 fragments. We can see here. And then based on this plasmid mass, what do you expect on fragment sizes that are produced? So in order to know that, we take the base pairs that we already have minus the ones, then the BAM-HI and the HIN-3 are taken away, which is 807 base pairs. So we put 807 here, and the remainder of the 5,512, which would be 4,705. And the same one goes for the para one. We take away the 377 base pairs. So we write this one here, and then the remainder is 4,495 base pairs. Which components will be found in each fragment? We construct the 4 fragments we left by typing the correct component in each fragment. So the fragments we would expect would be PBAD and the RFP, as well as the CAN-R. For the pKnR, and for the para, it would be the AMPR, the ORI, and the RC. And RC. And here we will put no gene present. So next, we're going to go read through the protocol. We're going to set the temperature of the water bath, set the volume of the P20 micropipette, drop the reaction buffer in the micropipette, pipette the restriction buffer into the K plus tube, pipette the restriction buffer into the K minus, A plus, and A minus tubes, dispense the other solutions into the K plus, K minus, A plus, and A minus tube. We're going to spin the solutions, incubate the solution tubes at 37 degrees Celsius, stir the solutions in the freezer. So first we're going to set the temperature. So I'm going to follow the instructions that are set here. Select the P20 micropipette from the micropipette rank, this one. I'm going to change the volume to 44 microliters. And then we're going to open the tip box and select the tip. We're going to close the tip box. So we're going to move the selection buffer, which is this one here, and open it. And we're going to use a micropipette and we're going to draw up the restriction buffer. We're going to close it. And then we're going to open the K plus tube. And we're going to dispense the restriction buffer by holding on the plunger till it reaches the second stop. We're going to close it, and we're going to inject the tip. And then we're going to close the K plus tube. We're going to do the same and add it to the K minus tube. Now we're going to do the same for the A plus tube. and the A minus 2 as well. Now we're going to follow the micropipette procedure again and dispense 4 microliters of the PK and R plasmid from the K tube into this back into the K plus tube. Okay. To the K plus tube. And follow the same procedure to dispense 4 microliters into the K minus tube. And then we're gonna dispense 4 microliters of paraplasmid A, which is this one, into the A tube with the A plus. And we're gonna follow the same for the A tube minus. Gonna dump the tip. Now you see this changed into 2 microliters. And now we're gonna dispense 2 microliters of restriction enzymes into the K plus tube. And we're going to do the same to the A plus tube. And now we're going to add 2 microliters of distilled water to the A minus tube and to the K minus tube. going to put the micropipette back. Next, we're going to select the microcentrifuge. Move the K plus, K minus, A plus, and A minus tubes into the microcentrifuge and balance them correctly by placing tubes at equal distances from each other in the microcentrifuge. Equal distances. We're going to check the balancing. Balance already, okay. So we're going to close the microcentrifuge and we're going to put the microcentrifuge to spin for 10 seconds. Once the spin is complete, open the microcentrifuge and move the solution tubes back. Then we're going to move the solution tubes into the floating rack. And we're going to close the microcentrifuge. So we're going to open the water bath, move the rack in here, close it, and set the timer to 1 hour. And press start. When the time is up, remove the floating solution tube rack from the water bath by pressing the eject button. Okay. Eject. Put them. Select the freezer and open it. Move your rack. Close the freezer. Now, the results. Once you have performed your restriction digest, you will not be able to confirm your results simply by looking at your tubes. In this image environment, we provide a hypothetical view of your results. Here are the actual results. So we're going to compare the actual results to the ideal results for the A plus, the A minus, the K plus, and the K minus. If the restriction digest was successful, you should see the plasmid DNA has been cut to linear fragments like these ones. Like the ideal results, which has the cut up plasmid DNA into linear fragments. So these are the LD results. As you can see, as the actual results are the same, because the A plus and the K plus are the ones that contain the cut up plasmid DNA, as we can see here, unlike the A minus and the K plus. In order to confirm the results of the restriction analysis, you will need to use a technique called gel electrophoresis. This technique uses a porous agarose gel to separate the molecules by size and charge. Having a good understanding of predictive firm size will allow you to correctly interpret if your enzymes cut the plasmids correctly and efficiently based on the size of the bonds observed. This is what I predicted for the digested K plus and A plus results. The 807 base pairs along with the 4705 base pairs, because as I mentioned before, we took away and put the remainder. The same goes for the para, the base pairs. And once you have run your samples through the gel electrophoresis, you can confirm whether you've produced the correct size fragments using the DNA ladder. The ladder contains DNA fragments of no sizes for you to compare with the sample bonds on the gel in order to estimate the size. If your digest was not set up at optimal conditions, optimal temperature, and correct precision buffer, you may see a partial or incomplete cut. The gel below shows the ideal gel electrophoresis one for the digest products. So as we can see, for the K plus, we have a 4,000 around between 5,500 base pairs and 4,000 base pairs. We can assume this is the one for the 4,000 705 base pairs, as well as the one around here, which would be the 807 base pairs, which would be this one. And compared for this would be for the pecan R, and for the para, we have the 377 base pairs, which would be way down here. And the other one, which would be the 4,495 base pairs, which would be this one. So as we can see, the predicted bonds seem to be correct because we see them in the DNA ladder here in the K plus and the A minus, A plus, my bad, K plus and A plus. Now that we complete the restriction digest, which tubes can we combine to create the new recombinant plasmids? Well, it will be the K plus and A plus tubes. Let's look at what you learned from the simulation. Please click on your answer to each of the following questions to save it in your notebook. Okay, so what do you think is the main purpose of this simulation? It would be to learn how to digest plasmids using the restriction enzymes. As you can see, this is correct. What is the purpose of heating the tubes 37 degrees Celsius? Because this is the temperature at which the restriction enzymes function best. Why did we add the same restriction enzymes to both the PKNR and paraplasmids? So that, which one is this? To create the same sticky ends on fragments from both plasmids that can be joined together later. Why did you need to spin the tubes after adding your solutions? This would collect all samples at the bottom of the tube so the reaction can occur. What was the purpose of the K minus and E minus tubes? These tubes were used as a control to show the impact of the restriction enzymes on the size of the uncut plasmid. Yep. And how confident do you feel about this technique? I feel very confident. So as you can see, all answers were correct for all the questions. And that's it for this lab. I will be reading the summary on my own.